Question 2 about earthworms. It gives us a little bit of information at the start. Earthworms live in soil. They feed on dead and decaying plant matter. They have soft, moist skin, and they exchange gases through a skin. Probably nothing you don't already know there, but anyway. Give 2 abiotic factors and 2 biotic factors that could affect the size of an earthworm population. So what sort of things might affect how many earthworms you have in a particular piece of soil? So first of all, we're thinking abiotic factors. So these are abiotic means non-living, so non-living factors. So things like the amount of water, the level of water, the level of moisture, humidity. Just the word water will do. If the soil is wet or dry. The amount of things in the soil that the earthworms might need. So minerals. Thinking about the pH of the soil, because soils vary in pH. So we could say availability, and also the food available for them to eat. So availability of course, no, sorry, so food would be a biotic factor because they eat dead plants and animals, but availability of minerals would be one. The pH level of the soil. Things like temperature as well. I don't think there's anything else. I suppose the nature of the soil. So is it clay soil or sandy soil? So that it's a bit general, but type of soil, something like that. Sandy or clay. Maybe the size of the soil particles, whether it's rocky, stony, etc. Then we've got biotic factors, so these are the living factors, and I mentioned food before, and of course food is, if they're eating, as it tells us at the top, they feed on dead and decaying plant matter, so we definitely have food, so dead plant matter, would probably be the biggest one. But we could also have, and this is sort of going to be the same for any biotic factors, it's things like predators, what's going to eat the earthworms, moles eat earthworms, all sorts of things like that, and then disease, so if there's diseases that are going to affect their population. So generally, for biotic and abiotic factors, they're generally going to be the same answer, you just might have to apply them to different situations. Question 2.2 is a 6-mark question. Not too bad a one though. Students investigated the populations of earthworms in the soil in 2 different areas. Area A, which is a grass lawn, and Area B, which is a farmer's field. So this is our independent variable. This is the thing that we're going to change deliberately each time we do the experiment. It's the area A, but it's the area that you're sampling. It tells us chemical X can be mixed with water and poured onto the soil. The mixture brings earthworms to the surface of the soil but does not harm the earthworms. So it's a way of counting earthworms without having to get down into the soil and dig them up and disturb the soil. So that's why they're talking about chemical X. They're not going into details about what it is, they're just telling us that it's a way of seeing how many earthworms are there. So plan an investigation using chemical X to compare the number of earthworms per metre squared in areas A and B. So our basic method is going to be to add chemical X to, now it's going to be, we're going to have a sample at a certain point in, let's start with area A, so we'll say 1 metre squared, 1 metre squared in area A. And collect and count just the number of worms. We can get to count the worms that come to the surface. We don't necessarily know we're going to have got every single worm that's under that 1 metre squared, but as long as we make sure it's a fair test, which we'll come to now, we can still compare the two areas. Obviously we need to make sure that the metre squares that we are testing are randomly selected, and we're not just going to do one in each area. We'll randomly select. And it's often good with these sorts of things to explain that a little bit. So using random coordinates. Five times 1 metre squared. And you could, or if you're using quadrats, it might be 0.25 metres squared, because a quadrat is normally 50 centimetres by 50 centimetres. But let's assume we've got a 1 metre square quadrat. Quadrats in area A. Okay, so then we've got a set of data on area A. We then just need to repeat repeat this process in area B, but crucially what we need to do here is explain our control variables. So using the same. So first of all, with chemical X, we need to use the same concentration and the same volume of chemical X. I'm sort of imagining it might be poured from a watering can or something, so you could even go on about, the method of applying it or something. But the key thing there is going to be the same concentration, the same volume of chemical X is used in each sample. The same sample area. So put it to into a comma. The same sample area, so it's got to be 1 metre squared each time. And the other thing would be, if you're putting this chemical onto the soil, and then you're counting the earthworms that come to the surface, you're going to have to wait a certain amount of time for the earthworms to appear, right? And we don't know whether that's going to be 10 seconds or 10 minutes or whatever, but as long as we keep that consistent, it's going to be a fair test. So the same sample area and the same, so it's the same time between application and counting. We've probably covered all of the key control variables there. Now obviously we've then got 5 data points for each area, area A and area B, so we just need to calculate a mean calculate the mean for area A and B and compare. So we're then comparing the means of each area that we've been that we're sampling in. Okay, so that's question 2.